day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Dita Makwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So, in today's lesson, we are looking at our grade 12 exam revision. We are going to be preparing for our final examinations. So today we are looking at DNA, the code of life, and we will be focusing on DNA replication as well as DNA profiling. So we're going to be looking at the process of DNA replication, whereby the DNA molecule makes an identical copy of itself, as well as the technique or process of DNA profiling, whereby we're going to be looking at DNA profiles, just getting a reminder of how to interpret these DNA profiles, as well as looking at the uses of a DNA profile. So what are we going to be looking at within these topics? These are some of the things that you must make sure that you know about the two topics before you go into the examination room. So firstly, the process of DNA replication. As we know here, you're going to know when in the cell cycle does the DNA replication process take place? So when does it take place? Secondly, where in the cell does it take place? So when and where does it take place, right? Next, how DNA replication takes place. So there we are going to look at the process itself. We're going to look at it from the beginning all the way to the end. Make sure that you know how to describe this process. Make sure that you know exactly how the process occurs, so that whenever you're asked a question about DNA replication, you can simply just describe it and get your marks. It's usually a 5 to 6 mark question. Make sure that you know that question, the significance or the importance of DNA replication. Why is DNA replication important? Why is it important for DNA replication to take place? Make sure that you know that as well. Right, so we are looking at two topics that are quite straightforward. They're very straightforward. You just have to make sure that you know everything here, right? Secondly, with DNA profiling, we have the interpretation of the DNA profiles. So you must be able to look at the DNA profiles and know exactly what is happening there, which is what we're going to look at, right? We're going to just remind ourselves on how to interpret this and how to use these DNA profiles basically, and then also to look at the uses of DNA profiles. What are DNA profiles used for, right? So we're just going to look at those as well. But remember, whenever you are asked a question on the uses of DNA profiling, let's say your DNA profiling question is on paternity testing, right? Now, if the question comes and it says, what other uses, name other uses of DNA profiling, it means that you cannot mention paternity testing because you're already looking at paternity testing. So when you come across that kind of a question, you should be able to list other uses other than the one that you have been looking at in the question, right? Now, the structure of the question paper, you should know exactly what kind of a structure are you going to be looking at, what kind of a question paper are you going to be looking at, especially because in the previous years we had a question paper that has section A, B, and C, right? Whereby the first two sections made up to 130 marks, and the last section was 20 marks, which was an essay question. We no longer have the essay part. We only have section A and section B. Section A is always 50 marks, right? So the section A has 50 marks and about 4 or 5 questions. The first 3 questions, it's 1.1 up to 1.3. 1.1, we have the multiple choice questions. 1.2, we have the terminologies or the biological terms. Now, with regards to these terminologies, make sure that you learn all your terminologies as you are studying. So you can start by going through the terminologies as you start with your studying or your preparation for the exam, so that as you are going through your notes, you know exactly what is being spoken about when they mention certain terminologies, right? And then the next one would have the matching column or matching items. So here we have the mentioning whether it is A, B, both A and B, or none, right? And then after 1.3, we usually have 1.4 and 1.5, which are short questions that are usually based on some diagrams that are provided. And then in section B, we have two questions here. We have question 3 and question 2 and question 3. Both of them are 50 marks each. So in question 2, you're going to have long questions as well. In question 3, you're also going to have long questions. Like I said, each question is 50 marks, which will then give us 100 marks. That 100 plus 50 marks would be 150. So 150 marks for 150 minutes. 150 minutes is the same as 2.5 hours. So your paper will be out of 150 marks for 150 minutes. Right. Now, by the end of our lesson today, you should be able to describe the process of DNA replication. That is one of the main things we're looking at here, describing that process of DNA replication. And then being able to interpret a DNA profile. Like I said, you should look at the DNA profile and be able to interpret what is happening there and be able to answer questions based on what is happening on the DNA profile. And then you should also be able to describe the uses of a DNA profile. What is a DNA profile used for? Like I said, if you are already looking at a question on biological evidence in a crime scene, for example, you cannot mention that. You can then mention the other uses other than the one that was provided in the question. So make sure that you know all these uses. Okay, now we're going to look at some examination tips. We're going to look at ways in which you can tackle the paper better, right? So we're going to look at the marking principles. The marking principles are basically just to guide you on what the markers and the examiners expect from you, right? Now, if you know what is expected of you, it means that you will be able to give that in your paper. As you are writing that exam, you will know exactly what is expected of you, and you will be able to do that as you are writing that paper. So firstly here, we're just going to look at the principles as I've said. So if more information than marks allocated is given, you stop marking when the maximum marks are reached. So for example, if you asked a question out of 2 marks, right? And then the information that you provide is about 3 or 4 marks. Once I reach the 2 ticks, I stop marking and I put the max indication to show that you have reached your maximum marks, right? Secondly, if, for example, 3 reasons are required and 5 reasons are given, mark only the first 3, irrespective of whether all or some are correct or incorrect. What this simply means is that you have questioned, they said give 3 reasons. Now you have reason 1, reason 2, and reason 3. Now, if you're looking at all these reasons, I mean, you give 4 and 5, you give more than 3. Now, if you're looking at all these reasons, this one is incorrect, this one is incorrect, and number 3 is correct. And then these 2 are also correct. Just because these 2 are correct, it doesn't mean I should then go on and mark these 2. I'm not allowed to mark these 2. I'm only going to mark the first 3 because the instructions simply said 3 reasons. Now, if you give more than 3 reasons, it means that you do not take instructions, therefore the remaining will not be marked. Number 3, if whole process is given when only part of it is required, read all and credit the relevant part. Well, that is an advantage. However, when you are used to writing more than you are required, you are going to waste your time. Remember, 150, 150 marks, 150 minutes. Now, you are not going to be spending a lot of time on questions that will not give you marks. Make sure you do not do that. Number 4, if comparisons are asked for and descriptions are given, accept if the differences and the similarities are clear. All right, now let's have a look at number 5. It says if tabulation is required but paragraphs are given, candidates will lose marks for not tabulating. So if the question says tabulate, make sure that you tabulate so that you get that 1 mark for drawing a table. Drawing a table gives you a mark. Now you do not want to be losing marks for simply drawing a table, right? Now next, if diagrams are given, with annotations. When descriptions are required, candidates will lose marks. Make sure that you follow instructions. That is the most important thing. Follow those instructions. It said describe, then it means you must describe in words. It didn't say give a diagram or annotations. It says describe with words. And then it says if flowcharts are given instead of descriptions, candidates will lose marks. Again, do not give flowcharts. Describe in the form of words. Number 8, caption. All illustrations must have a caption. When we're talking about illustrations here, we are talking about the graph, the tables, the drawings, and diagrams, as it says. So they must all have this caption or heading to make sure that the person who's going through your work knows exactly what you have drawn, right? There is also a mark for that. And then number 9, if only the letter is asked for, but only the name is given, and vice versa, candidates will lose marks. Make sure that you give that as well. If it says name and letter, give both the letter and name. If it says letter only, give the letter only. If it says name only, also give the name only. Don't waste your time giving both, even though it is not expected. Right. And then here now we are looking at the general tips. However, here I just want us to look at, especially for this lesson, I just want us to look at learning of terminologies. Make sure that you learn those terminologies, like I said earlier. Make sure that you know the terminologies, because it's even easy for question 1.2 in the exam, because you are given the terminology, the description, and you must tell us what that paragraph or that sentence is describing. So simply knowing and then giving the name of the part that it's is being described makes it very easy. So make sure that you do that, right? Eliminate the incorrect options when answering multiple choice questions. For your multiple choice questions, make sure that the incorrect answers, the answers that you know are definitely incorrect, are eliminated. Then you remain with the ones that you are not too sure of. As you are comparing, it will make it easier for you because you have eliminated some and you are only left with one or two. Right. And then always use a ruler when comparing the black bands or the bars on a DNA profile. So now, this DNA profiles, remember, are going to be compared based on where the black bars or the black bands are going to be found. Now, based on that, you are then going to be able to say, okay, because both these individuals' bars have been found on the same place on that DNA profile, it means that they share some type of genetic material. So it is very important to be able to compare them properly by using a ruler. Okay, now again, when you are looking at DNA replication, remember that both strands serve as a template, whereas in transcription, only one strand serves as a template. So make sure that you know that DNA replication is different from transcription. In DNA replication, both those strands are going to serve as templates because we need them for the production of new DNA. Whereas in transcription, we only need one mRNA, therefore the DNA, only one DNA strand can act as a template. All right, with the terminologies, we have DNA replication, the process by which a DNA molecule replicates to produce an exact copy of itself. That's the process we're going to look at. And then the cell cycle, that is a series of events that occur during the lifespan of a cell. So those events that make up the cell cycle are also taking place for a reason. For example, we have interphase, right, during which certain um, processes such as DNA replication takes place there. Now, it is important for that to take place. And then all the other events also, as they're taking place, it is important for them to take place. A template is a strand of DNA upon which another strand of DNA is built during replication. And then lastly, what is a DNA profile? These are the black bars that are left behind on an X-ray film when an extraction of DNA undergoes a biochemical process. And then we have DNA profiling, the process of identifying an individual by comparing his DNA with another known DNA. That other known DNA will be the DNA that has been found on a DNA profile. Okay, we've come to the end of our examination tips. Let's look at the definition of DNA replication. Where does DNA replication occur and when does it occur? Right now, you should be able to define this. As I said, know all your definitions, know all your terminologies, right? So, this one, it's the process by which the DNA makes an exact copy of itself. Right. So if in the examination question 1.2 you get this description, the process by which DNA makes an exact copy of itself, you are simply going to give the answer and say DNA replication. That's it. Now, where does DNA replication occur? Remember, it occurs within the nucleus because that is where we find our DNA, right? That is where we find our DNA molecule. Right. And then when does it occur? It occurs just before cell division begins. What do we mean by cell division? What are we referring to when we are talking about cell division? We are talking about mitosis and meiosis. Remember, we have interphase, which takes place before mitosis and meiosis, and then we can start saying mitosis has started or meiosis has started. Right now, in meiosis we have the other phases, right? We have prophase, metaphase, anaphase, and telophase. Now, this interface is where DNA replication takes place. That's what we mean when we say DNA replication takes place just before cell division. This is the cell division part, right? The mitosis and the meiosis part. That is the cell division part. Now, replication takes place before both mitosis and meiosis, before cell division can take place. So it occurs during interphase, which happens before mitosis, before meiosis, in preparation for these two phases. I mean, these two processes, right? So DNA replication would then take place inside that nucleus. We have that DNA that replicates itself, and then you are now going to end up with two DNA molecules that are identical to each other. Simply like making a photocopy. When you make a photocopy, you have your original, you make a copy, and it comes out exactly like your original, right? So it takes place in the nucleus in preparation for mitosis and meiosis, right? Now, why is it important? What is the importance of DNA replication, the significance of DNA replication? We already know that this is for the DNA molecule to make a copy of itself. Then now, why is it important for it to make these identical copies of itself, right? So, especially during mitosis, is that when the daughter cells that are going to be produced, remember mitosis produces two daughter cells, and these two daughter cells are supposed to be genetically identical to each other. Now, if we look at DNA replication, it produces DNA molecules that are genetically identical. Mitosis produces cells that are genetically identical. Now, the DNA, the genetic material, is supposed to be found within these two cells, is also supposed to be genetically identical. Therefore, it means that it is important for the genetic material or the DNA produced by DNA profiling, DNA replication to be shared amongst these two cells so that the two cells can also be identical, right? So the reason for DNA replication is to make sure that the daughter cells that result from mitosis each contain the identical genetic material. So they must have identical genetic material, as we can see. Now, if DNA replication does not occur, we would have this cell. This cell will not be able to produce these two cells. However, because of replication, mitosis starts with this kind of a cell. Now, this kind of a cell is now going to produce these two cells, which are now genetically identical. This is only due to the process of DNA replication, and without it, we wouldn't be able to see that. Now, remember, these genetically identical cells are going to be diploid, as we can see, and they're going to have the same number of chromosomes as each other and as the parent cell. Okay, so it also ensures that the two new cells at the end of mitosis contain the same amount of DNA as each other and as the parents. Now, during meiosis, we have DNA replication during interphase, and then we have meiosis 1 and meiosis 2. Unlike in mitosis, here we have 2 rounds of the cell division, which is the meiosis, and we have 4 haploid cells like this. From one cell, we're going to have 4 haploid cells. Now, these 4 haploid cells are going to have half the chromosome numbers. If DNA replication had not occurred in order to double the genetic material, it means that the genetic material that was going to be shared was not going to be enough for the 4 haploid cells. And also, in this case, we do not expect the DNA material to be genetically identical at the end of meiosis because of the process of crossing over, which makes sure that the cells are genetically different from each other. Right, now let's have a look at the step-by-step of this process. We start off with this DNA molecule. Remember, before it even gets here, it will be a helical structure, a double helix structure that is twisted. Now, that twisted structure is going to unwind. When it unwinds, it's now going to look like what we are seeing here. It's going to form this ladder-like structure. Now, once that ladder-like structure has formed, we are going to see that the weak hydrogen bonds holding the nitrogenous bases together is going to break. So the weak hydrogen bonds are going to break. As the weak hydrogen bonds are breaking, the two strands are going to separate. This DNA molecule is going to unzip. As it unzips, the two strands are going to separate. Now, when the two strands have separated, each of these strands are going to be used as a template. So each strand serves as a template. This strand is going to serve as a template, and this strand is also going to serve as a template. When we have these templates, the free nucleotides, remember, nucleotides are the building blocks. Remember, when we speak about building blocks or the monomers of a DNA molecule, we talk about those nucleotides. A nucleotide, remember, is made up of a phosphate, a sugar, and a nitrogenous base. Now, this nucleotide is going to come in, and then the nitrogenous base of that nucleotide is going to attach to the nitrogenous base of the template of the original DNA molecule. Remember, not just any nucleotide is going to attach. Guanine will attach to cytosine, thymine will attach to adenine. So we are going to have what we call complementary base pairing here. The free nucleotides, the free DNA nucleotides will attach to the original DNA templates, and we're now going to be seeing the formation of the new DNA molecule. Free nucleotides with matching nitrogenous bases line up next to the exposed bases. These are those exposed bases in the original strand according to their base pair rule. The base pair rule, thymine to adenine, guanine to cytosine. Right. Now, we are going to see that they are going to attach until we have that long chain, and that chain is now going to form the DNA. So the new deoxyribose and phosphate molecules join to form a chain alongside each strand of the old helix. So we'll see that as they are attaching there, we're going to start seeing the new DNA molecule that is going to form. So the hydrogen bonds will reform. As the hydrogen bonds are reforming, it means that now the two DNA molecules are going to be attached by those hydrogen bonds. Now you will see that you would start seeing the DNA structures. There we have one DNA molecule and another DNA molecule. And these two DNA molecules, as you would see, they are going to be identical to each other. What tells you that they are identical to each other? Let's have a look. If you have a look here, you would see we have CG and we have CG. Because of the complementary base pairing, that is what allows these two DNA molecules to be the same. If we go through everything here, you would see that they are all the same. They are all the same until we get to the end, you would see that we have these nitrogenous bases that are bonded to each other exactly the same. Now we have two DNA molecules. One DNA molecule is made up of one original strand and one new strand, right? One original strand and one new strand, and then we have two DNA molecules. And remember, the process of DNA replication is controlled by enzymes. Now here we're going to look at the process again, but I just want us to look at it with the structure in mind. Now, if we have a look at the structure, you can see that it is going to guide you on what happens at each step. Step number one, the main thing here is for you to mention that the molecule unwinds. So this twisted molecule is going to unwind to form that ladder-like structure, and then the weak hydrogen bonds break. When the weak hydrogen bonds break, the DNA strands are going to unzip. That is the second step. So that is where you're going to be getting your marks. So no matter how much you explain, no matter how much you write, but if you're not writing the main things, if you're not writing that the DNA strand has unwinded, if you're not writing that the weak hydrogen bonds have broken and the strands have unzipped, you will not get those marks. Make sure you mention these important keywords. Right. Now that they have unzipped, it means that they have separated from each other. Right. Now, each strand, now they are separate. Each of these strands are now going to serve as a template. This is why we are seeing step 3 twice happening on both sides. This is our template and this is our template. And then from there, you will see we have a template, the template on both sides, especially on the outside. And then on the inside, we're going to see the formation of the new strand. So there we have our new strand. There we have our new strand. But on the outside, what do we see? We are going to be seeing the old strands. There we have our old strand here and our old strand there. So now we can see that there's going to be the complementary base pairing again, whereby adenine pairs to thymine and guanine pairs to cytosine, right? Complementary base pairing of the free DNA nucleotides from the nucleoplasm. And then at the end, you will have two identical DNA molecules, one with one old strand and one new strand, and the other one also will have one old strand and one new strand. Remember to mention that the process is controlled by enzymes. Okay, now guys, when you describe these kind of processes, make sure that you, even if you forget some parts, make sure that you write down whatever part you can remember. Make sure that you do not miss anything. If you miss something, continue writing whatever you can remember. As the marking guidelines said, if it is a description, everything will be read and the relevant parts would be awarded marks. So make sure that, right? Even if you don't remember the full process, at least in the end, mention that we have two DNA molecules that are identical and the process of DNA replication is controlled by enzymes. You can score your 2 marks there. Maybe even at the top you have remembered that the double helix unwinds, then you would get a mark there. Just make sure that you do not leave your question unanswered. Do not leave any blank spaces when answering questions. Write whatever you can remember. Okay, let's have a look at our first activity. It is activity 1.4. It says study the following diagram showing DNA replication and answer the questions that follow. Okay, obviously they've already told you that it is DNA replication. It means that there's no question where they're going to ask you what process this is, right? Okay, now if we have a look at this, it is a 6-mark question, right? So I'm going to give you 6 minutes. Why am I giving you 6 minutes? So that you can practice to answer the questions in the allocated time. 6 minutes for 6 marks. I'm going to give you that 6 marks. Quickly go through it. Attempt to answer all these questions as best as you can, and then we'll come back and discuss them. All right, now let's go through the questions together. It says, identify the parts labeled 1, and then the sugar 2, and then 3. So let's go back to our diagram and have a look at those. So we have part 1, we have 1 here, we have the sugar over there, and then we have the whole thing, which is 3. Now, at 1, remember, we are looking at a diagram, which is this part. We're looking at a nucleotide, the building block of a DNA. What makes up the building block of a DNA molecule? This question is guiding you so much that you should not even miss a mark here. Why? Because already at 2, you have been told that it is a sugar. What kind of a sugar do we find in DNA? That takes us back to the previous lesson whereby we learned about the difference between DNA and RNA. The sugar for DNA is a deoxyribose sugar. Right, now at 1, what makes up a DNA a nucleotide, a DNA nucleotide? 3 things. The sugar, we've already been told that it's a 2. This part we can already see that it's a nitrogenous base. It means that now there we are left with a phosphate portion, right? And then 3 is talking about the whole thing. 3 would be now the nucleotide, the DNA nucleotide. All right, so we have the phosphate, the deoxyribose sugar, and the nucleotide. All right, and then the next question, it says, give 2 reasons why the above diagram represents replication and not transcription. Now, when we were looking at those tips, I did mention that we are looking at DNA replication, and when looking at DNA replication, you must remember that each strand of DNA acts as a template, or both strands of the DNA molecule act as a template. And also, we know that during transcription, when transcription occurs, we are looking at the formation of an mRNA molecule and not another DNA molecule, right? So now, if we have a look at this diagram, we can see that there are going to be 2 new DNA molecules formed, and the 2 strands are actually acting as our templates. That would be our first reason. The second one would be that the nucleotides that we are seeing here are not RNA nucleotides. How do we know that they are not RNA nucleotides? RNA does not have a thymine nitrogenous base. We cannot see thymine nitrogenous bases in RNA, but we would be seeing uracil. But in this case, we can see thymine pairing to adenine and not uracil. So there are 2 new DNA molecules. Remember, in transcription, only 1 mRNA is going to be formed because only 1 strand serves as a template. And then there is no uracil, there is a new thymine base complementing the adenine. Okay. 1.4.3, give 1 significance, give 1 importance of a DNA replication. We looked at the importances. We looked at the fact that during mitosis, it needs to maintain the chromosome numbers so that the chromosome numbers remains the same. Because remember, the cells that are formed at the end of mitosis should remain the same as the original one or the parent cell. Therefore, everything must remain the same. The chromosome numbers must remain constant. Without DNA replication, the chromosome numbers would reduce to form identical cells. The cells that are going to be formed at the end of cell division are going to be identical as well. All right, now our second activity, it says describe the process of DNA replication. Now, here you've been told to describe the process of DNA replication. In another question, it would say describe the process during which DNA makes an exact copy of itself. In that case, you are not told that it is DNA replication. It means that it's either before you describe the process or at the end of the process, you must then mention that the process is DNA replication. When you do that, you will then get a mark for that. But in this case, you do not need to mention it again because it will not be a mark. All right, so here again, let's just take a few minutes, 5-6 minutes again. Write down that process, describe that process, mention all the keywords that we have spoken about, mention the unzipping, the unwinding, the templates, the complementary base pairing, and at the end, the fact that you end up with two DNA molecules that are identical, and the fact that it is controlled by enzymes. Okay, 5 minutes and then we will discuss it. All right, now let's have a look at the answer. It says here the double-stranded DNA unwinds. Now, again, you can see that the ticks are not just placed at the end of the sentence, but they are placed in certain parts. It unwinds and unzips, right? There is a mark there. The hydrogen bonds break. You get a mark for that as well. And then each DNA strand is used as a template, mentioning that each serves as a template is a mark. Now, if you say one of them acts as a template, you will lose that mark to form a complementary DNA strand. When you're talking about complementary DNA strands, you can also explain this part whereby adenine pairs with thymine and guanine pairs with cytosine. How does it do that? It does that using free DNA nucleotides from the nucleoplasm, and then two DNA molecules that are identical are going to be formed. Like I said, this part you do not mention because you will not get marks for that. However, if the question did not mention that it's DNA replication, you would then mention that this process is called DNA replication, and then it will also be a mark. If it is not mentioned, it will be a compulsory mark, meaning that if it said 7 marks and you did not mention DNA replication, you would be marked out of 6. So make sure you do not miss that. Even if it has been mentioned, you can also mention that it's DNA replication. Then the person who's marking will just leave it out. To be safe, you can always mention it. Again, you should also mention that the process is controlled by enzymes. All right, now let's move on to DNA profiling. Here, we are going to be looking at mainly the identification of individuals using these DNA profiles. We can identify different individuals using their fingerprints. Now, these fingerprints have some problems in identifying individuals because due to where they are found, they can be altered by surgery, by wearing gloves. There is ways in which you can work around them in order to make sure that your fingerprints do not appear, right? So all of that makes the use of fingerprinting to not be reliable in identifying criminals, especially. However, if we look at a DNA profile, we are using a DNA of an individual. The DNA of an individual cannot be altered, mostly because, especially because the DNA is found in every cell is exactly the same. The DNA of an individual is identical throughout the body. Why? This is because the cells in your body are produced by mitosis. Cells that are produced by mitosis are genetically identical. Therefore, they are exactly the same throughout the body. So your DNA cannot be altered. It means that should you leave your DNA at a certain place, that DNA will be compared to your DNA and it will remain the same, regardless of where the DNA you left was found and where the DNA is being compared to is found, right? Okay, so it is very useful in identifying individuals. This may not be in terms of crime, but even in identifying relatives and so on. We're going to look at some of the uses of this DNA profile. Now, what is a DNA profile? A DNA profile is the diagram that we are looking at here. Now, this DNA profile is made up of dark bands. These bands here may be looking white because they are appearing on a black background, but they are usually very dark bands, black bands or dark bands that are left behind on an X-ray film. After DNA had been extracted and undergone the biochemical process. Once it undergoes this biochemical process, we're going to see these indications by the black bars, which would represent your unique DNA. Now, if we take two individuals who are not related at all, their DNA profile will not appear like this because they do not have the same genetic material. So this profile will appear a certain way based on your genetic material, right? Okay, here we are going to look at the interpretation of a DNA profile. Like I said earlier when we looked at the tips, bring your ruler to the examination room. Without that ruler, you will find it difficult to interpret this. Now, with your ruler, you are going to put your ruler across and it is going to be able to check which black bands or which bars are matching with each other. Remember, by comparing the black bars, you can be able to identify whose DNA that you are looking at, where the DNA belongs to. Basically, let's have a look at this example here. We have a victim who was stabbed, and we have found the knife from the crime scene, which contains some of the DNA from the suspect. Right. So here we have the victim's DNA, we have the DNA from the crime scene, and we also have the suspects, their DNA profiles. Now we're going to compare all this DNA and to see which suspect had committed the crime. So as you are comparing the profiles, you are going to use a ruler to just indicate which of the suspects have the black bars or the black bands that match the most with the one from the crime scene. Then the one that has the most black bars matching to the ones from the crime scene would be the one that has most likely committed the crime. Now we can see here the first bar belongs to suspect number 1. The second bar belongs to number 2. The second one, suspect number 2 and 1. And then the third bar belongs to the victim, suspect number 1, number 2, and number 3. So this one belongs to everyone, matches with everyone, not belongs, but matches with everyone. And then the fourth one, it matches with suspect number 2 again. The next one matches with 1 and 2. The last one matches with 1 and 2 as well as the victim. So now, if we count the bars that matched the crime scene, we will see that all of the bars from the crime scene have matched with all of the bars from suspect 2. Then we can say that suspect 2 is the one that has most likely committed the crime. And you should also know that by saying that this sample matches with that of the suspect, it does not necessarily mean that or automatically mean that the suspect has committed the crime, but it means that they were at that place during the time of the crime. Right. Now let's have a look at the uses. We have the diagnosis of disorders. This can be used to diagnose disorders, the development of cures. So once you have diagnosed a disorder, you can use that information to also develop cures for the disorders. Biological evidence, mostly here we're looking at criminal cases. And then lastly, identification of relatives. It can be relatives, it can be paternity testing, it can be deceased individuals. Right. Now, with the diagnosis of disorders, we can look at disorders such as hemophilia as well as the sickle cell anemia disorder, which can be diagnosed in newborn babies as well as in unborn children. Right. And then, like I said, with the information that has been obtained as the diagnosis have been done, also the use of the DNA profiles can be used in research for these inherited disorders, right? So the researchers can use DNA profiling to find patterns in large groups of people with or without the disorder. So as they are looking at these patterns, they will be able to understand how the disorders occur and also try to find ways in which they can get the cures for this, right? So the understanding is necessary in the finding of cures for the disease. And then biological evidence in forensic science, we can be able to use this to find criminals, right? So as we have said, it is a reliable method because your DNA cannot be altered, right? And again, we know your DNA does not look the same as anyone else's DNA, but you should remember this does not tell us that the individual committed the crime, but just tells us that this individual was uh, found in the place of the crime at the time the crime was um, committed. So here we can have your blood, we can have your semen stains, your hair that was left at the crime scene, some skin cells also. Let's say you were involved in a fight and the victim scratched the suspect, the skin cells can be used to obtain the DNA of the person who committed the crime there, and that can all be used to create a DNA profile. So every part of your body that contains your DNA can be used to extract your DNA in order for the DNA to be used. Right. Lastly, identification of criminals. Like I said here, it can be used to determine the father of the child. It means that it can be used for paternity testing. It can be used to trace siblings. So if two people suspect that they are siblings, their DNA profiles can be created and compared. Obviously, when we compare the DNA profile of siblings and we compare the DNA profile of parents and child, it will not be the same. If it is from parent to child, then the matching bars would be more than the bars of the siblings. However, it is going to show some matches if individuals are matches, right? Are siblings. And then identifying people who have died, and cannot be recognized. It means that we can use these DNA profiles to compare to the DNA profiles of their relatives, their family members, in order to make sure that it is the person that is suspected to have died. Now, we're going to look at the interpretation of two DNA profiles that may not be exactly the same. So we have the semen, meaning that we're looking at crime here. We have the victim and we have the suspects, just like the previous one that we've looked at. And then we have this one whereby we are looking at paternity testing. We're going to start with the one with the rape crime. So here we will see that the semen matches with the bars of the victim, right? And then it also matches with the bar of suspect 3, right? So suspect 3 and the victim. The second one matches suspect 1 doesn't match because it's a bit thin. It matches suspect 3 again. And then the next one is very thin as well. Now, as you can see, I'm finding it a bit difficult to see if the victim and the semen are matching with this small band. That is why I said bring your ruler. Make sure you have your ruler. Now we can see it matches a bit with suspect 1 as well and suspect 3. The next one matches suspect 3 only. Next one again matches suspect 3. Once again, suspect 3 only. And then here matches 2 and 3, and for 2 it's quite thick, so it is not a match. Lastly, it matches suspect 3. So now this tells us that this semen is belonging to suspect 3, so that can tell us that suspect 3 could be the rapist. Right, now with paternity testing, here we have the profiles of the child, the mother, and the possible fathers, 4 possible fathers. Now, when you do a paternity testing profile, you're going to start by comparing the profile of the mother and the child. Why is that? Remember, this child is going to have genetic material, 50% genetic material from the mother and 50% genetic material from the father. Now, let's count the bars of the child. 1, 2, 3, 4, 5, 6. It means that 3 bars should match with the mother and the remaining 3 bars should match the father. Now, if 3 bars match the mother and only 2 bars or less match these possible fathers, it means that they are not the fathers, because both parents must match the child. There shouldn't be any bar of the child that does not match with any of the parents, right? So whichever male here is going to have 3 bars matching to all the remaining, not the ones that have matched the mother already, but the remaining ones. is the one that would be the father. Now let's start with that. The first bar of the child matches with the mother. So it means that we're going to exclude that bar when we match with the fathers. The second bar of the child does not match the mother. It means it must then match the father when we are comparing with the fathers. The third bar matches the mother. I've ticked it. It means that when we match with the fathers, we do not look at it. Fourth bar doesn't match the mother. Bar number 5 matches the mother. It means we are not going to look at it. Bar number 6 does not match the mother. How many bars are matching the mother of with the child? 1, 2, 3. The remaining bars must match one of these males. If it doesn't, it means that none of them are the father. Right. Bar number 2, let's have a look at it. There we have it. B and D. Right. And then bar number 4, it matches A, B, and C. Right. And then bar number 6, it matches B only. So now bar number 2, B, bar number 4, B, and bar number 6, B. It means that the possible father here would be male B. All right. Then the remaining ones cannot be the fathers because they did not have 3 bars, all the remaining bars of the child matching to them. Right, let's have a look at activity 3. It says the diagram shows the DNA profiles of 6 different people. We have individual A up to individual F. Now, it's a 6-mark question, 6 minutes. You go through the questions and then we are going to discuss them. All right, now the first question A, it says give the letters of the 2 people who are identical twins. How do you know that 2 people are identical twins? Their DNA profiles are going to be the same. Only identical twins are going to have DNA profiles that are exactly the same. Now, already we can see that these 3 are exactly the same, and these ones are also exactly the same there. Right. Make sure that you use your pencil to identify the individuals that are matching, because you cannot just have a look at it and decide that they are matching. Okay, now A no longer matches with B and C, so we can exclude A as a twin to B and C, right? Because A is no longer matching. And then we were looking at C, E, and F as those that are matching, but now we can see that C is no longer matching with E and F, but E and F are still matching with each other, right? And then we have a look again. Let's have a look at B and C. B and C are no longer matching here. However, let's have a look at some more. Okay, we can see that C is still actually matching with E and F. Let's have a look at that. C is matching at E and F. We're not comparing B and C because initially they were not matching. Let's go back and just erase that part. Here, we cannot compare B and C. Let's erase this as well. And then let's go back. C is still matching E and F. There, we have them. They're matching the second part, the first part, and the second part, they are still matching. The third bar of C, C is matching with F only. It's no longer matching with E, right? Then we move on to the next bar of C, is matching both of them. But because the previous bar was not matching, we can already exclude E as the twin to C, right? And then this one is matching with F again, and then the last bar is matching with F as well. So we can conclude that bar 1 are matching, the second bar matching, the third one matching, the fourth one again is matching. So we can conclude that individual C and individual F are indeed identical twins. right? So C and F are our identical twins. And then the second question says, give the letters of the parents of person B. Right, so it means that you are going to go back to the profile again and have a look at person B, right? It means that we must erase this. Use a pencil, not a pen, because then all of what you have done would disturb what you're trying to do. So use a pencil so that you can erase and now focus on individual B. Individual B must have two individuals who matches them. Okay, now let's have a look at individual B. So firstly, these two bars of individual B are matching both A and D. So we're going to just put a cross on both A and D, right? And then we're going to look at the second bar, which is this one. Who does it match? It matches C, E, and F, right? And then again, the next one, it matches E only. The next one, it matches A only. The next one, it matches E only. And the last one matches both A and E. Now, we're going to count the number of bars that are matching with all these other individuals. F will be excluded because at least half of the bars of B must match with F. So F is not the father, is not the parent. And then D also only one matched, not the parent. C again, not the parent, because only one bar matched as we are looking at them. Now, if we look at A and E, the remaining ones, we can see that 1, 2, 3 have matched. And then again here, 1, 2, and 3 have matched, or we can say 4 have matched. Now we can then say that individual A and individual E are the parents of person B, right? So A and E are the parents of person B. And then C says, is the collection of DNA from every citizen in South Africa to create a DNA profile database for South Africa a good idea or not? Explain your answer. So would it be a good idea to have a DNA profile database? That would be a good idea because so it means that you can say yes and explain your answer. That would be a good idea because it would be so much easier to be able to identify individuals while comparing them to the unknown DNA profiles. You just take that unknown DNA profile and compare it to all the DNA that you have, and that would make it much easier to compare DNA profiles, unlike going around trying to find suspects and then getting their DNA profiles first and then comparing. So that would be a good idea. However, you can also say that it is not a good idea because something like this would mean that it is going to invade the privacy of individuals, right? So let's start with the no. So the DNA profiles may reveal personal information about a person, personal information that people do not want to reveal, which could be used against them in a prejudicial way, right? Or if you say yes, you can say that it can be used to identify crime suspects as well as relatives and also assist in organ transplants. Remember, once you have these profiles, you have a lot of genetic information about that individual. So it can help in a lot of ways as well. Now, just to summarize what we went through, DNA profiling, DNA replication, the DNA unwinds, the double helix unwinds, and then it unzips and separates. And then the two strands are going to serve as templates to form the new strands using complementary base pairing, where adenine pairs with thymine and guanine pairs with cytosine. And then remember, both strands serve as templates in this case. And then we're going to have the two new molecules that are genetically identical, and the process is controlled by enzymes. And then with DNA profiling, the uses here, parentage or paternity testing, the medical diagnosis of disorders, or finding cures for these disorders. It can also be used in the food industry as well as in agriculture. And lastly, forensic science, whereby it can be used to help solve these criminal cases. Now, make sure that you bring all of these things to the exam room with you, because this might be one of the reasons why you fail to answer some questions. If you have a calculation question, and you do not have a calculator, you could simply lose marks because you don't have that calculator. If you are required to draw a pie chart, make sure you have this. You can never, ever, ever, ever draw a correct pie chart without the compass and your protractor, right? As I said, your ruler. This can be used when you're drawing your graphs or your tables. As I've said, the interpretation of your DNA profile. So make sure you bring that, your pen, your pencil, and your eraser. Always bring those because it will just make your writing process so smooth and easy. All right, in the next lesson, we will then be looking at the process of protein synthesis. We will look at transcription in the nucleus and translation in the cytoplasm. And we're going to be looking at everything that could happen here, the mistakes that could happen in the DNA molecules, in the formation of the mRNA, and the roles that could play in the formation of the protein. All right, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.